We're here at the 21st CROI with Romas Geleziunas, who is the head guy of the what we will call the towards a cure research, I guess is the best way to put it, the work that would lead to a cure, hopefully. And this is a big challenge, and I think before we really get into any of tedium, I think we really need to talk about the dynamic challenge that this is to the massive undertaking. If you can kind of capsulize that, that would be a great way to start.
Yeah. Well, first of all, thank you for having me, Fred. It's always a pleasure to visit with you. So thank you very much for having me. Yeah, so this is, it's quite a fantastic enterprise that is being built, and this is in academia, industry. I mean, the partnerships that we're seeing being formed are quite unique and exceptional, I think, at least for the history of HIV. We've never seen this before. So the examples are, for example, DARE and CARE collaboratories, the Defeat HIV collaboratories, you know them well. I mean, these are consortia of outstanding scientists with some industry representation. And so these are individual collaboratories that are working together. I can tell you that we have numerous collaborations underway as well with government, and I can get into some detail later. With other industry partners, we're starting to develop relationships. So it's quite a fantastically big enterprise now, and it's exciting because it brings together a lot of smart people with different expertises that we will need to basically take on this challenge of eliminating reservoirs of HIV. So it's really a very, very big enterprise right now.
And your company has been invested in well, it started out with just the antivirals, and then you were looking for those best suited to do a good job for you there. But what I guess when we realized that there was an opportunity for a cure, then that focus was made very prominent in your business plan. So I think that to me is a sign that not just your organization, but so many have done that to feel not just be a part of it, but really there is a serious effort to find a cure. Do you find a way in which you could kind of tell us a little bit about how a scientist goes about the work that they're doing? Give us a day in the life of, if you will, of the work of a person who's doing this.
Well, let me give you a day in the life, or let me back up and give you an idea of how we actually do this, because the day in the life, my personal life is not that exciting because I actually read and write emails all day and speak to people. So that work per se is not exciting. The exciting stuff is what the scientists are doing in the laboratories and the clinical folk who are trying to bring these concepts and treatments to the clinic. So, but to give you an idea of how this operates, first and foremost, the biggest challenge that we have in industry is understanding what the target is, because that's how we operate, right? For example, in the case of HIV, we tackled reverse transcriptase, the enzyme that turns the RNA of the virus into DNA, integrase, protease, right? These enzymes that HIV has. So once you have a very concrete enzymatic target like that, you can go about setting up assays and then to screen compounds to inhibit those enzymes. That's straightforward and direct now in retrospect. In the case of HIV cure, the challenge is we don't fully understand the target. The target being, what are we trying to achieve? We know we're trying to achieve the elimination of reservoirs that persist during therapy, but we don't fully understand what those reservoirs are. That makes it extraordinarily complicated. I mean, the one reservoir that we do understand and agree upon is this resting memory T cell that we can obtain from the periphery of patients. So we can isolate this from a patient. We can then put it into tissue culture in the laboratory, and we can then apply agents to see what kicks out HIV, what would kick out and then kill those cells. But I think, for example, at CROI, we're starting to realize that there are mysterious other reservoirs that we don't understand. So by not even knowing what the target is, we have a tremendous challenge. How do we go about tackling or eliminating that reservoir if we don't even fully understand what the full spectrum of reservoirs are? So that's what makes it very, very challenging. So that's why a lot of the effort that we have spent, and at Gilead we have been working on this for about 5 years now, has been spent on trying to activate latently infected memory CD4 T cells and trying to kill them, obviously, kick and kill, the strategy that we all talk about. So that's where we've spent a lot of our time. So we've devised assays to try to mimic those cells that are amenable to high throughput screening. And so we've screened our compound collection, commercial libraries, et cetera, to try to find initially, for the first several years, agents that would activate latent HIV. And that's how we spoke about this last year, we discovered the drug romidepsin as being particularly effective at activating latent HIV. So this is only a small part of it, yet it's huge. Already what I just described is many people's work over many, many years. And that's just the front end of this. We have not yet identified several candidates that we'd like to nominate for clinical testing. We've not found those candidates yet. We've not found candidates that we think are suitable for that. So,
have you when you come to a conference like this, you get a lot of cross information. The basic science that comes along. And does anything jump out at you and say, oh, like Michael Malam's presentation with the APOBEC3 and so forth? I mean, that's been going for a little while. I think he and others discovered that 4 or 5 years ago. Does any of that come to mind as maybe being helpful, or that might lead you down a pathway that you needed to be led down? Or
yeah, that's a terrific question, because that's the primary reason we come to these conferences, because what we're trying to do is to better understand these targets, these reservoirs, if you will, and trying to see whether we could what type of research we could basically use to basically arrive at a better understanding of reservoirs and identify agents that would eradicate them. Yeah, Michael's presentation was terrific. He's a terrific scientist. Obviously, everybody knows that. In his area of APOBEC, as you can imagine, there have been numerous companies that have tried to identify inhibitors that basically will inhibit the interaction of VIF with APOBEC. Now, that falls more in the realm of the antiviral space. Right now, what I and several of my colleagues who work on HIV eradication at Gilead, what we're looking for is novel ideas in terms of reservoirs and strategies to eradicate them. So I'd like to maybe mention the work of Matthias Lichterfeld. He showed in his presentation that there is potentially a new reservoir, which is this memory stem cell. And these memory stem cells give rise to the central memory and the effector memory cell, and that basically comprises this latent resting reservoir that we have been studying. So now there's a new player potentially upstream of those guys that could give rise to those latently infected memory cells. So we have to think about this carefully. That may represent what I was mentioning to you earlier, a novel target. That suddenly gives us a concrete thing to go after. So can we isolate enough of those cells from patients? Can we devise model systems that could mimic that? And can we now study, again, screen compounds, test? Exactly. So those are the types of things that I look for. What new targets, what reservoirs, what kind of reservoirs should we be thinking of beyond memories, resting memory T cells? And strategies like those.
Well, now the collaborations and collaboratories, are they? You all are meeting and you talk about where you're at. Does everybody kind of get on the same page, and then you kind of work? Somebody may not believe this isn't particularly important to them, but you believe it is important, so you work on it. And I mean, everybody kind of knows what everybody's doing. There doesn't seem to be this cutthroat competition.
Oh no! Thanks for mentioning that.
Which I think we brought up last year, but I think it bears repeating.
Oh, absolutely. Yeah, that's long gone. I mean, it absolutely does not exist in this area. Everything that we do is collaborative. Everything. The Forums project, Veronica Miller's project, is reuniting all the scientists that work in this area to try to define regulatory paths for these cure therapies once they arrive. The IAS group has an industry collaborative group as well. I attended a meeting this week during CROI, and again, reuniting scientists from academia, from government, and from industry. The Annenberg Foundation is creating also a forum to study that. So, I mean, virtually everything that we do now is collaborative. Everything, and the reason is very simple. This problem is so daunting. I told you, we're struggling understanding fully the targets that we need to go after. It's just so daunting. So we need to put the best and the brightest minds together, and we need to be talking all the time, sharing data, and trying to advance this cause as fast as we can. And it'll only be done if we're talking and collaborating. And I see that it's the new reality. It's our reality, and it's fantastic because I can I just get on the phone and call up colleagues, and we can start discussing things pretty quickly. They can do that with me. So it's...
And their insight might be very empowering to your thought.
Absolutely, absolutely. The best way to achieve or arrive at solutions is getting a bunch of smart people in the room and start a debate, a discussion, and challenge each other, and that's how solutions arise. So we definitely have that going on in the field right now.
Now you have a lot, like you said. There's already a host of bright scientists that have jumped in here because they're already in HIV and they migrated to this more specific area. But the other thing I'm thinking about is that we do not an interview, but a panel with the young investigators each year. Each year I see these extraordinarily bright-minded people, and we beg to the people in the audience that are extremely smart to get into the field, because it seems that you have people like yourself, or especially that are much older, who have this, we've been there and done that, but you didn't have this knowledge here now. So you get the people the young folks who are going through school now are getting a lot of new stuff, that their repertoire of knowledge is collectively different than yours or someone who's much older. And maybe your thinking pattern is going to be different. This is my feeling, that getting bright young people who are not maybe ready to dismiss something because we tried that and it didn't work, now you can retry it based upon the new information.
You're absolutely right. I mean, fresh blood from young people, from folks outside of the field is very important. It's definitely going to help us. I can tell you, not only young people coming up with new ideas, but also folks from the oncology field. This problem of reservoirs that we're trying to address is somewhat related to what folks in oncology are facing. This is a persistent cell. It doesn't expand, but it persists in the body. So the types of strategies that folks in oncology are using, we're trying to borrow some of those ideas to bring them to HIV and try to use these strategies to kill a persistent, long-lived cell. So, so we invite and discuss this question of reservoirs with folks in the oncology field. So beyond what we just discussed, these massive collaborative networks within the HIV cure scientific community, we're reaching out to other scientific communities to bring their knowledge into the picture. So again, this enterprise is pretty huge right now, and a lot of hardworking people are trying very hard here.
We had a gentleman, I can't remember his name now, from Perth, who did a little bit of work with Microsoft and with Nebojsa Jojic, I remember his name, on the vaccine work that they did. And they had this giant, how they were going to custom design some sort of drug or a compound for each person, because it would be specific, and I know that didn't seem to fly well in the face of others. And there's always a lot of competition, I shouldn't say competition, but disagreement within scientists about, you can't do that, or it's too complicated or whatever. But would it be helpful to, and that was in vaccines, but do you believe that there's any way for maybe the scientists, the technology scientists to come to play here, and computers that can answer a lot of questions for you very quickly? Or do you already have that engaged?
Well, we have some of that obviously engaged at each of our companies, particularly in the biopharmaceutical industry. We have such departments, and so we obviously engage them when needed. But absolutely, I mean, we're keenly watching any type of development that could help this cause. So we obviously, and the beautiful thing is when you reach out to folks and you explain what the cause is, people are very receptive. They come immediately. Yeah. Oh, absolutely. They come in and like, if it's for HIV eradication, absolutely. What can I do? And so it's really easy to get folks to engage and collaborate on this. Right. I'd like to mention, though, you mentioned the vaccine question. This is kind of the holy grail. If we had a therapeutic vaccine, right, that we could, inject maybe once or twice or 3 times and achieve eradication, that would be fantastic, not only for us here, but in Southern Africa, right, where this problem is quite daunting and huge.
There's a huge number of people living with this disease.
Exactly. Yeah. So if we had, the dream would be, can we develop a vaccine, that we can inject just a finite number of times and eradicate this virus? So we've not been very successful in the area of therapeutic vaccination in this field, but there are some new ideas that are coming around, and one that I want to mention, and again, you'll see why, because it's another nice collaboration. So I think I mentioned this last year when we spoke, Louis Picker's vaccine, right?
Oh, yeah.
Presented that last year.
The last presentation at the CROI.
Exactly.
And
exactly.
all the smart people stuck around for
Exactly. Well, because it's an important breakthrough. And I think we had discussed that a little bit. So the concept is he uses Cytomegalovirus, which is a herpes virus that most of us are infected with. It's benign in our bodies until we become immune suppressed. Then it would cause some pathologies. But in most cases, we live with this virus. So basically, he has used this virus and dropped into this virus, some, in this case, SIV antigens. And down the road, of course, the idea is to use this for HIV vaccination. So he put HIV antigens into human cytomegalovirus. But for now, what he has done is taken monkey CMV and he put SIV antigens in there and vaccinated monkeys, and he achieves this remarkable result, 50% protection of all animals that are challenged. And he has a series of papers that he has published to follow up on this since last year. It turns out that the monkeys that are protected, so the vaccine does not block acquisition. The monkeys get infected, but they have the ability to control and eventually clear. That is, eradicate, eliminate that virus from their bodies. So this is an important development because it says that there's a way to instruct the immune system to combat an AIDS virus, an aggressive AIDS virus. It's a monkey AIDS virus, but it's an aggressive AIDS virus. So you have a way to instruct the immune system to combat this virus and get rid of it. That's an important blueprint right there. We need to really, focus on that and make sure that to understand whether that might be helpful in therapeutic vaccination. So what we have done is a beautiful collaboration between the Gates Foundation of Philanthropy, Louis Picker's group, Jeff Lifson's group at the NCI, and Gilead. And we came together. We're going to collaborate on basically testing precisely that concept. So we're going to do the monkey study first. So SIV infect the monkeys, treat them with antiretrovirals for a prolonged period of time, vaccinate the animals, and then release ART and see if that vaccine can confer control like it did in the prophylactic setting, but could it do it in a therapeutic setting? That's a very important study in my mind. So anyway, groups came together to get this done.
Of course, I'm proud to be in the same city with Louis Picker in Portland, Oregon, as that's where our organization
is.
That's
right.
So I know we talked to, his name Sasha, I think it was. I can't remember in Malaysia last year, and of course he's working in this field, and a very bright young man, and like I say, it seems though the brightest people gravitate toward the biggest challenges. I mean, if you're a bright person, you want to solve a real big puzzle. You don't take on tiddlywinks. You've got to do something that's really complex.
I agree. I mean, engaging the young folk that you were talking about earlier in this problem. I mean, again, it's very easy. It resonates. Do you want to participate in trying to find a cure for AIDS? It's hard to say no to that. If you're a young person, that's an exciting challenge because you know how important it is and how much good it would do for mankind. So it's easy to engage folks. Exactly, it's a no-brainer.
Well, I appreciate the work you do, and I'm trying to figure out if there's any other areas that we should cover, because you mostly work in this, like the vaccination, you're working on that, and the cure. But is there any other novel approaches that you might be looking at that you'd care? I mean, I know we don't want to bring something up before it's time, but that you think might be worthy of putting out here? Yeah,
I'd like to mention one other item that we've become excited about, and again, it's a study that was done in monkeys, and it's a study that was done by Dan Baruch, a colleague of ours, and it's about these broadly neutralizing antibodies. So these are antibodies. In Dan's case, he was using antibodies that were cloned and identified by Dennis Burton, IAVI, and a small company in Seattle called Theraclone. And so this group basically identified and cloned antibodies from elite neutralizer patients that had sera that were broadly and potently neutralizing. So they identified individual monoclonal antibodies from humans. And so Dan Baruch's study, what he did is he infused these types of antibodies, and one in particular, PGT-121 was particularly effective, into monkeys, and he saw a very profound and abrupt suppression of plasma viremia. Something very, very dramatic. And in a small group of animals, after 1 or 2 infusions of this antibody, he saw sustained plasma virus suppression. So we have not seen that before. So this obviously, again, looking for clues as to how to treat or eliminate these reservoirs, we're asking ourselves, could these types of broadly neutralizing antibodies, could they have benefit in addition to antiretrovirals? Could they do things that antiretrovirals don't do, which is to maybe eliminate or attach to these reservoir cells and maybe find a way to kill them by some effector mechanism that is governed by the immune system. So we're keeping a close eye on that as well, and we're working hard to, again, create another collaboration, speaking to various groups to try to get a key study off and running, and that would be combining this type of antibody with antiretrovirals and see if we can achieve suppression in larger numbers of monkeys, sustained suppression in the absence of antiretrovirals. So the idea would be here, combine the antibody for a finite duration with antiretrovirals, and then stop the antiretrovirals, stop the antibody, and see if the monkeys can have sustained suppression. And with that concept in mind, you can think of translating this to the clinic because it's a human antibody. It came from a human. So that concept could translate quickly if the data were promising in monkeys. So another exciting concept. So there's ideas are floating around right now.
So the other thing that jumps to mind is that we have to be thinking about, I think we always thought as one drug was going to make the virus go away, initially, way back at AZT a million years ago. And now then we came up with multiple. And then how are we thinking about deploying? I know there's a cascade of events that has to happen in like you're saying, you have to bring the virus out of all the compartments and then kill it, that concept. And then there's also the issue of how much of it is going to take to do that. I mean, maybe you have several compartments. One drug is going to do this in this compartment, another drug is in one, and another drug doesn't cross the blood-brain barrier and has to. So you get all this going. So do you have that sense of how that might play out, or is it too soon to decide how much stuff we're going to have on board to make the whole picture work?
Yeah, it's a very good question, Fred, because I don't think you'll find anybody in the field who'll say, we're confident that one agent will take care of this problem because of what I said earlier. Likely multiple reservoirs residing in different places in the body, different types of reservoirs. So it's highly likely, and it makes good sense to think that you're going to need a multi-pronged combination approach of reactivation of virus when the virus is present in cells in a deep coma. You have to bring it out. If you don't bring it out, that particular cell might be dormant for 8-9 years and might eventually pop up. So it'd be best if we could draw those cells out during a period of treatment and flush those guys out. There are sanctuaries. There are places maybe that certain cells of the immune system might not be able to get to. I can think of several groups that have shown us that there is a reservoir potentially of infectious HIV in T follicular helper cells. And these are cells that reside in germinal centers where CD8 cells, the CTLs that we think could be helpful in eradicating HIV infection if they were effective, or effective enough, they don't go to these germinal centers. So they're excluded. So even if you had a great vaccine that elicited a great CD8 response, maybe those CD8s will not migrate to the site of this reservoir. So you have to think of alternate strategies. So I think it makes great sense to think that a combination,
maybe a sequential or a sequence or
combinations
or whatever the process is. But You're thinking, I guess you're always thinking outside the box. Thinking, how can we make this work? What sequence we can do, or what process, or what combos, or so it's a lot of deep thinking, I guess, on this program.
And the other aspect is which ones of these strategies can we bring to the clinic sooner? They're not at the same stages, obviously. There are inherent challenges with all of them. I mentioned the antibody approach. I think that one has a really good shot of making it to the clinic if the data in the monkeys, warrant it, suggests that there is a strategy there that could help us reduce reservoirs, because we know a lot about human antibodies and we have the ability to make them. So on the spectrum of things, that has a greater chance of success earlier on. Some of these other strategies, the activating agents, we have to be more careful, with them. We have to understand them very well. So these agents, I think, will arrive at the clinic at different times, depending on safety profiles, which is, our number one concern. We don't want to harm anybody. That's the number one thing.
And the ethics and how to inform consents and all that. We talked about that at the NIH last summer, I guess.
Exactly. The risk benefit. What is it?
Yeah, and that's part of what we say now to the audience, is that they're going to be times where we're going to ask them to invest altruistically in some work that's going to not necessarily give them an immediate response or maybe never a response. It may not be necessarily beneficial to them specifically, what we're doing, but it will be beneficial to the knowledge that we have to proceed down the line. This is not something that's going to be next week or next year. It's going to be down the line. This is a long-term commitment we're asking people to think about, not to jump to conclusions, not to say, okay, the baby was cured, so we're taking our kids off treatment. I mean, this is crazy. We have a process by which and the doctors have to follow, and the patients have to follow, and the parents of kids have to follow. It's a very serious bit of work, and we need the support of the community in so many ways, and the media for not jumping in the gun and saying, oh, it's all over. You know, as soon as something happens, they say, may this be the end of AIDS today, you know, and no, it won't be that easy. As you described.
That's very well said, Fred. You captured it beautifully here. You're absolutely right. Setting expectations clearly, and no, we don't have a cure for AIDS. It's not for tomorrow. You're absolutely right. You really said that very, very well. I do want to mention another thing that we have done with the community that I think is very, very important. You know, we've devised a lot of these assays for high throughput screening, but ultimately, when we find agents, we have to validate those agents in patient cells. And we have this long-standing collaboration now. You and I both attended this community pre-CROI meeting together, and Jay Lalazari was there. We have this long-standing collaboration with Jay Lalazari and his patients. And it is because of that collaboration that we were able to identify romidepsin and really generate data that was, I think, very robust, that led that data in the patient cells from Jay's clinic led to the ACTG study in which we're going to test romidepsin in HIV-infected patients on suppressive ART. So this, again, another collaboration. It's a company, Gilead, working with a private clinic and patients from that clinic to try to identify strategies that are worth testing in the clinic. So I thought that was a beautiful, again, another example of beautiful collaboration, the types of collaborations we need. So
and the usefulness of the patient. And
absolutely.
I mean, when you're asked to do something like that, and it may be a person who's not infected or a person who is infected, we need all volunteers that are willing to do that opportunity to do with the. I mean, I call a lot of the doctors heroes because they really are working really hard, almost tirelessly, at this question, and it's an obsession. I know it is with a lot of the activists too. There's a lot of activists, people living with AIDS that will really be a part of the cure, I'm sure, at some point. My part of it is getting the message out, and I hope that this is helpful.
Yeah,
I think it will be. I think
absolutely.
We covered a lot of good territory here.
Yeah.
Okay.
No question.
Thank you very much.
Always a pleasure.
Romas, thank you.
Thank you.